Conference 2020. This is the third session. It is a web conference organized by Lee Katz, Andrew Page, and myself, Nabil Ali Khan. And I have to give you the fine print that this conference is not sponsored by CDC or Quadram Institute, and the opinions expressed here do not necessarily reflect those of CDC or Quadram. We are pleased to have all of you here today. I should mention that the Zoom session will be recorded and made public. So if you don't want to be videoed, please close that now. And also, please remember to keep your audio muted. We really hope to go beyond the standard webinar and have a lot of social mixing. So that's a really critical part of the meeting in MeetSpace. And I hope we can translate that here. So that interaction is driven by the Discord server running alongside these Zoom meetings. So please sign into that as well. The link is in the group chat of the Zoom session. And we'll move over there afterwards to have a virtual coffee break where we'll mix and talk and ask more in-depth questions of the speakers and talk some more science. And I also invite anyone to join me in for a virtual beer or coffee if you're attending, if it's the middle of the day. So today's session is going to be a mixed bag of evolution, clinical microbiology, and some population genetics. We will have some very exciting talks from Conor Meehan, Leo Martins, and Rebecca Bengtson. I hope I said that right. And so first up, we have Conor, who's got his slides all ready to go. And Conor is a lecturer in molecular microbiology at the University of Bradford. His research focuses on evolution and epidemiology of microbes, primarily pathogenic mycobacteria, such as Mycobacterium tuberculosis, ulcerans, and leprae, with an emphasis on whole genome sequencing coupled with a phylogenetic-based analysis. So over to you, Conor.
Great, thanks very much. So start this off here. Hopefully you can see my screen there.
Yep, it's all good. Good.
Yep, good to go. Okay, so today I'm going to talk to you about some work that was done in a large collaboration between 3 different groups where we found a new lineage of Mycobacterium tuberculosis. So I could just walk you through what that means from the Mycobacterium side, but I thought it might be interesting to show how we define a new lineage, all the different checks and balances, so that we're not just talking about a random clade from a phylogeny, but really about something that is distinct and worthy of the title lineage. So I like to start off with my acknowledgements, so you don't think that I do any of this work by myself. Especially this work was done in a very large collaboration. These are the main people that were involved, but there's a couple of dozen people overall that had some influence in one place or another. So we have the ITM Belgium, where I used to work before I started here in Bradford, with Bauke and Lane putting a lot of effort in here. In Rwanda, my PhD student, Jean-Claude, is doing a lot of work there. In the Swiss Tropical Institute, we have Chloe and Sebastian, and then the University of Lille, we have Michel and Philip. So for those who don't know too much about it, Mycobacterium tuberculosis is the causative agent of tuberculosis, which is a primarily respiratory disease. We have about 10 million new cases every year, with about 1.6 million deaths every year. So this averages out to about 4,500 deaths every day from tuberculosis worldwide. A lot of this is concentrated in specific countries. You can see on the map here on the right that we have most of it occurring in sub-Saharan African and here in Southeast Asia, but we do have TB cases in every single country in the world. So it's spread primarily through aerosols, close contact between people. We have a long latency before active disease. A lot of the time you have maybe a year or 2 between contracting the disease before you'll start to show symptoms, although only about 1 in 10 people will actually progress to a symptomatic stage who have tuberculosis. So tuberculosis has been around for a long time in the human population. This is a Bayesian tree where the internal nodes are inferred ancestors of these different parts of the tuberculosis population structure, and the years prior to present of when they are inferred to be around. And you can see that all of the TB has been around for probably about 3,000 to 4,000 years, but some of these estimates go up to 16 or lower, maybe down to 2 or 3. It's been found in old Peruvian mummies. It's been found in religious sites around different parts of the world. and all this kind of thing. So it's a disease that's been around for a long, long time with a large population structure going back hundreds, if not thousands of years. So a little bit about that population structure before I get into the lineages. So Mycobacterium tuberculosis is caused by a collection of strains which are referred to as the Mycobacterium tuberculosis complex, the MTBC. This historically has been named multiple different species like Mycobacterium tuberculosis, Mycobacterium africanum, bovis, caprae, oregus, microti, pinnipede, and other things. But a paper that came out a couple of years ago showed that these are all one single species using different techniques like average nucleotide identity and digital DNA hybridization, all these things we use to define a species. And in bacteria, the definition of a species is actually quite well, let's say, resolved genomically. There are certain things you need to tick off in order to define a species. So all of these individual species got renamed into variants. So this whole complex, the species, Mycobacterium tuberculosis complex, evolved from an environmental organism which is similar to Mycobacterium kineti. So kineti is the closest organism that we know to the rest of these Mycobacterium tuberculosis. So inside of this complex, everything that was called a species before is now called a variant, especially Africanum. So now we have Mycobacterium tuberculosis variant Africanum. Although if you go to NCBI, some of them have been renamed, like bovis is now a variant, but for some reason oregus isn't. Taxonomy is never straightforward when it comes to anything bacterial. The word variant actually doesn't have an official taxonomic standing. Only everything from species and above, and sometimes subspecies, has a specific taxonomic meaning, supposedly, where things like variant, ecotype, complex, lineage, all of these are kind of free to be defined by whoever is writing that paper at the time. So within this complex, we tend to talk about 7 human adapted lineages. We have 1 here, 2, 3, 4, 5, 6, and then a new one from Ethiopia called 7. And then we have a lot of animal adapted ones, mainly listed at the bottom, like pinnipede, microti, bovis, caprae. But you can see that the human and the animals are not completely separated. The chimpanzee version actually sits in between lineage 5 and lineage 6. And we've had human ones, human cases of pinnipede. So these lineages have been defined primarily based on phylogeny. They are clades that are monophyletic inside of this phylogenetic tree, and specific large sequence polymorphisms, which are called regions of difference in Mycobacterium tuberculosis nomenclature. So we have them numbered here like RD9, RD7, RD8, and 10. These define different splits in this tree, how these lineages were originally defined. And these splits tend to be quite old. Lineage 2, lineage 4, the split between them is several thousand years ago. So not all of the lineages are found everywhere in the world. We have what's called modern lineages and ancestral lineages, with the modern ones like lineage 2 and 4 being quite geographically widespread. You can see the red dots all over the map here at the top being where we find lineage 4, which is basically every country that we find tuberculosis. Lineage 2 is also found in a lot of different countries, primarily around Asia. We have ones that are called intermediately distributed, found in multiple different parts of the world, but not everywhere, mainly lineage 1 and lineage 3 that are kind of around the Indian Ocean. And then we have some that are very geographically restricted. Lineages 5 and 6, which are also used to be called Mycobacterium africanum or West African 1 and 2, they're primarily only found in West Africa. You will not usually find anything from these lineage outside of this area. Lineage 7 has only really been found in Ethiopia or people who had traveled to Ethiopia recently. So even though we have a lot of tuberculosis in the world, it is not evenly spread amongst which lineage we find. Each of these have slightly different characteristics, some relating to virulence, or for example, lineages 5 and 6 actually tend to grow in microaerophilic better than they do in aerophilic, like the rest of them. So this is to say that we have 7 lineages overall that are currently defined, mainly defined upon phylogeny and these specific regions of difference that we see in different parts of the tree. So I want to talk a little bit about how we define a new lineage. So as this, Nabil said in the intro, I work a lot on tuberculosis, but I also work a lot on bacterial taxonomy for some reason. Sometimes I hate it and sometimes I love it. You see new lineages pop up all over the time with bacteria. You see this a lot in viruses. You may have seen a paper recently that split the SARS corona 2 virus into 3 different lineages based on some phylogenies. And it is it's very not clear about how to define a new lineage and what it should be. So new bacterial lineages tend to be primarily based just on phylogeny. Things that you would want your lineage to be are monophyletic, so everything in the lineage forms one clade or grouping on a phylogenetic tree, exclusive to everything else that's not in that lineage. We want multiple lineages to be about equally divergent. You don't want one lineage that has everything is very closely related to each other, and another lineage where everything is extremely dissimilar to each other. And you want something that's statistically supported, such as like a bootstrap, where we know on the phylogeny that this clade, this monophyletic grouping, is well supported on that tree and not due to misplaced root of a tree or specific lack of phylogenetic signal in the alignment, which means we don't have enough diversity to tell us that a strain goes in one place of the tree or the other. So this is what it should be for a new bacterial or even viral lineage. What we would desire is also something that's quite different from a current lineage. The split happened quite a while ago, and these populations tend to still be extant. So it's not a small couple of strains that are found in a few patients, but never were transmitted onwards. We wouldn't normally call that a new lineage because it did not create a new population structure. We'd also want another type of distinction. So even though I'm big into phylogenetics, I also believe other things should come into consideration when we talk about lineages. We would want some kind of maybe phenotypic difference between these lineages, maybe different virulence. As I said, some grow microaerophilically, some grow aerophilically. There's some examples of phenotypic differences. We might want genomic differences. So you can think of different subtypes of salmonella with different plasmids and antibiotic resistances. We don't have plasmids or lateral gene transfer much in Mycobacterium tuberculosis, but in other species, bacteria, we might want to see large genomic differences in terms of insertions, deletions of large sequence portions. Geographic difference also is normally something that's accounted for in lineages. We find this in one part of the world and this other lineage in another part, or environmentally in terms of the hosts that they infect, with some maybe infecting animals, domestic animals, others infecting humans, others infecting, I don't know, amoeba or something like that. We'd also normally want multiple strains to have been sequenced. So for example, I worked on HIV during my PhD, and there the lineages have distinct ways of defining them, and you had to have found multiple copies of the same subtype, as they're called there. You would want to have sequenced at least 3 different genomes in order to say that this is a circulating subtype. So these are the things that we'd like to tick off when we're looking for a new bacterial lineage. Although the vast majority of lineages that you will see out there are primarily based just on phylogeny, with a lot of these other desired things not taken into account. So I'll tell you a little bit about the story about how we came about finding this new lineage. And it started with what we call a spoligotype. So spoligotypes are a way of genotyping Mycobacterium tuberculosis complex strains. And it's based on the presence absence of CRISPRs inside of the genome. It's just called spoligotypes in Mycobacterium tuberculosis because they like to have different names for some reason for every single thing. So spoligotyping is the presence absence of 43 of these different CRISPR spacers in the genome. And it creates this 1-0 pattern. So you'll have 43 different places where you would look normally with either, you can do it with sequencing, or we have a kind of hybridization method on a membrane. And then if it's present, we give it a 1. And if it's absent, we give it a 0. And visually, as you can see over here on this side of it, we fill in the box black if the spacer is present and white if it's not. And then specific patterns can define certain lineages or sublineages. An example is if you look here at the lineage 2, a massive amount of the spacers are missing, with only spacers present at the end of this series, this would normally show us that it's a lineage 2. But there are a lot of different patterns that we see. It's not always consistent. You see that here in lineage 4, we have a lot of different patterns. The only thing that's consistent is this absence about, let's say, 4/5 of the way down. But in lineage 1, those patterns tend to be a little bit all over the place. This used to be used a lot for transmission. If two things had the same Spalago type pattern, you would say they're in a transmission cluster. But a couple of years ago, myself and some collaborators did a paper that showed that this has very low resolution for looking for recent transmission. So a collaborator, Sebastian Garneau, who works in Basel at the Swiss Tropical Institute there, in December 2017, he emailed basically a lot of different people saying that they were working on this project for Mycobacterium bovis at the time, and in a public database they came across this Spalago type pattern, which is very unusual. So there's no Spalago type pattern out there which has spacers at the start, and then none for these middle ones where we would hybridize, and then has some at the end. So this massive absence in the middle after finding these few present at the start is very unusual. So we emailed out asking, has anyone happened to see this pattern ever? Most people didn't respond or said, no, we've never seen it. At the same time, a new diagnostic trial called DIAMA had started, which was run by the ITM where I was working in Belgium, headed up by Bouke de Jong and Dessou Afolabi, who works in Benin there, where they were evaluating a lot of different diagnostics for Mycobacterium tuberculosis in Africa, one of which was what's called the duplex MIC-TB, which uses a targeted amplicon sequencing on a Illumina machine for specific genes in the genome which are related to drug resistance. And it also happens to give you the Spalago type of whatever you were looking at, so that you can know a little bit about the lineage of your strains. So that project actually started late 2017. Sebastian sent this email at the start of December 2017, and Philip Suppli, who was primarily the person who developed this duplex MIC-TB, as part of the DIAMA, had just taken some strains from Rwanda, where my PhD student Claude was in charge of it, just took a couple of random strains from multi-drug resistant TB patients and ran it through the duplex, and happened to have gotten the same pattern, or very similar, where we see these absences in the middle and we have some present at the start. So you see at the end it's a little bit different. So this was very much a fate thing almost, because this pattern had never been seen before, or really recognized in any database. And then two people happened to have come across it within the space of literally about 3 weeks. So this piqued Sebastian's interest, especially because he works a lot on the population dynamics, population structure of tuberculosis. Philip as well, who works a lot on Mycobacterium kineti and on tuberculosis itself, and my interest because I love the phylogenetics and population structure of tuberculosis as well. So the first thing we wanted to know was, where do they sit? Are they a lineage 1? They actually were put in the database, public database, as being bovis related, even though they were not, because they were not much is known about them, but that's what it had been labeled as. But it's not a Spalago type pattern that's ever seen in bovis. So the first thing we wanted to know was, where are these strains in this MTBC structure? So both of these happen to have Illumina reads done. For duplex, we sequence the whole genome along with the duplex results specifically for this. And for the other strain, which Sebastian had found in Uganda, there was already Illumina reads available in the public database for this. So we mapped these reads against a reference genome for tuberculosis, called the SNPs on that. We then basically gathered representative strain whole genome sequence data for all the 7 Mycobacterium lineages and all the different animal associated lineages, and also for Mycobacterium kineti, which was, we were acting as our outgroup here. We create a maximum likelihood tree using MaxML for this. It's a SNP alignment from all this data. We use an ascertainment bias correction here, where we put back in the invariant sites so that our branch links are corrected. We're using the GTR and CAT model here with 100 bootstraps to give us some support. We place M. kineti as the outgroup because we know that this should be the outgroup to Mycobacterium tuberculosis complex. And we get a tree here on the side. So if you're on a small screen, I apologize for being small. We have the normal structure that we would expect here at the top, lineage 2 being close to 3, and 4, 7 here. And our two strains sat basically at the base, or branching off from the rest of the MTBC, close to M. canneti. So this initial, based on the Illumina data, looked like we had a new lineage that was sitting between M. canneti, which is the closest extant ancestor to the Mycobacterium tuberculosis, and all the other lineages. So this already gives us a hint that we have probably a new lineage. It is not closely related to any other lineage that's there, as it forms its own branch outside that. We have two different isolates, so we know it's not a sequencing chimera or something weird that's happened somewhere. The two of them are about 200 SNPs apart from each other, so they are not highly closely related to each other. They are probably a couple of decades divergent from each other. So we have something here, and now we have to deep dive into it. The Uganda lineage, we only have what was on the public database. We tried contacting the authors. We didn't really get a response to try to get a hold of the strain. But we have the Rwanda isolate. And what we do then is we're going to go ahead and try to do some more analysis on that to not just say we have a new phylogenetic group, but we truly have a lineage that's of interest, perhaps clinically, phenotypically, or something like that. So this is where a lot of different studies would stop. They would say we have a new phylogenetic lineage, but we wanted to confirm that it definitely was a Mycobacterium lineage and that it had some properties that were of interest. So first things first, with a new lineage, we always want a new genome. So what we did was we took the Rwandan strain. It was isolated in 2017 from a 77-year-old HIV negative resident of northeast Rwanda on the border with southwest Uganda, where this patient had spent quite a lot of his life. And the other sample also had come from where we think is southwest Uganda, so the one that was in the public database. The strain is multi-drug resistant, meaning it's resistant to rifampicin and isoniazid, the two primary drugs used for treating first-line treatment of Mycobacterium tuberculosis. Because it's multi-drug resistant, this already gives us an indication that it's been transmitting in human populations, since it's been exposed to and become resistant to the drug. In fact, the Ugandan strain also, based on its genome, was also multi-drug resistant to these two drugs. So both of them were MDR-TB, meaning that we have human-to-human transmission going on of this lineage. It does not seem to be an animal-associated one, or not, at least not environmental. The Rwandan isolate was then grown up and gDNA was extracted using a method we developed for specific long-read sequencing extraction. We developed it in the ITM so that we make sure that we can break through the waxy coat of Mycobacterium tuberculosis. We sequenced it on a PacBio at Philip Supply did this. We assembled it using Canu, circularized it with Circulator, got very good coverage across the genome, and CheckM told us that we had a good completeness and we had 0% strain heterogeneity and 0% contamination. We used a pipeline called Magda, which annotated the genome for us, which had been used previously for Mycobacterium canneti. Again, from Philip Supply had done that. So I wanted to show you here, this is a duplex output on this side, these circles, that basically tells us what's resistant and what's not. That's where we can see the resistances and the weird spot type pattern in the middle. And this is just a nice picture of the colonies that we see from Mycobacterium, which gives us kind of rough edges, which we would expect from something from the Mycobacterium tuberculosis complex, whereas something like Mycobacterium canneti would look much smoother under a microscope. So a problem with phylogenetic placement of lineages that you need to ensure that the outgroup you use, in this case Mycobacterium canneti, is truly the outgroup when you've put in a new lineage that is very close to it. So what we want to do is expand the outgroup so that we can confirm that we're using it and that the routing is correct. So the question is, what if this one actually sits outside of Mycobacterium canneti? So if this new lineage was actually more basal, with canneti and the rest of the MTBC forming a group. So we wanted to make sure that that wasn't true, and what we did was we basically used a couple of other Mycobacterium species, which we knew were not related and should not be in this clade at all, would not have genomes that look like this. So some work that I was lucky to co-first author with people in Italy on, we did this huge phylogenomic structure of the entire Mycobacterium genus, and we found that indeed we have Candida and the other mycobacteria here in gray, but things like leprae nearby, Kanzasi, and Marinum and things like that. So we took the Kanzasi and the Marinum genome. We didn't take the leprae one because it's very, very reduced. It's a much smaller genome, so we wouldn't have much of a core genome to work with. But we took Kanzasi and Marinum, and we can use them as a extra outgroup so that we know that they definitely should sit outside of anything that we're looking at. We use Rory to create, we don't use a SNP alignment because we're looking at multiple species. We use an 80% similarity cutoff, which is what we had done in this paper in Rory to get a core genome. We create a super alignment from that where we concatenate all these core genes together. And we use Raxamel NG to create a tree similar to how I'd shown with the other one. So our lineage was basically confirmed to have the placement that we saw from the SNP alignment. This is based on closed genomes. So the one we had sequenced on the PacBio, and closed genome representatives of all these. We were able to have an expanded outgroup to confirm that our lineage sits here at splitting off a sister to the rest of the MTBC, but inside from Mycobacterium candida. So we're pretty happy that we have this intermediate lineage between candida and the rest of the MTBC, but we don't have anything on top of that. So I'm just going to talk a little bit about the candida. So what we wanted to know then is, it sits in between, do we actually have a new candida or a new Mycobacterium tuberculosis? These are the different things that we use to look at candida and Mycobacterium tuberculosis in terms of expected characteristics. M. candida is mainly found in the environment, and it's an opportunistic pathogen of people primarily in Djibouti. Mycobacterium tuberculosis is almost primarily human, although we have animal associated. Morphology is different. In the niacin test, which is a specific one for this group, candida would be negative, tuberculosis would be positive. Candida is highly divergent with a lot of ongoing lateral gene transfer within this species, a lot of SNPs between any two candida strains. Tuberculosis much more highly related to each other with no ongoing lateral gene transfer. This COB-F gene is related to B12 synthesis, is present in candida and absent in all of the MTBC. They cannot create their own B vitamins. We have this virulence gene PEPGRS35, which is absent in candida and is one of the things that's associated with the higher virulence of tuberculosis. And this PKS5 gene has 2 copies in candida, but a single recombinant copy in tuberculosis. So these are the things we would want to look for to see if it's more candida-like or more tuberculosis-like. So our new strain we've shown, because it's MDR, we know it's in human. We found it in a human as well. Morphology looks like tuberculosis. Niacin production is like tuberculosis. Diversity also looks like tuberculosis. No ongoing lateral gene transfer. We looked at this with clonal frame ML to look for recombination. What was interesting though is that it has the COB-F gene, unlike tuberculosis, much more like candida, and everything else like the PEPGRS and the PKS look like tuberculosis. So it's very like the other lineages of tuberculosis, but it's clearly an intermediary because it still has this COB-F gene, which is absent in the rest of it, meaning it probably can make its own B vitamins. So to finish up, I wanted to show you that we have a new lineage that resides between candida and the rest of the MTBC. We have clear placement and separation from the current lineages, which we showed with multiple different rooting, and we know that it's MDR-TB pattern. The genomic and phenotypic features are mostly like the rest of the MTBC, but it's retained this COB-F much more like candida. We need to now look into what the virulence of this would mean and the metabolic effects of this new lineage, as it's clearly some kind of intermediary in the evolution towards what we see much more widespread in the MTBC. We only found it in where I have this red box at the bottom, which is around the Great Lakes in Rwanda and Uganda. So we need to now search to see if we can find it in any other countries. We looked through all the public databases, whole genome sequencing, spaligotype ones. We did extra sampling in DRC Congo, and in Uganda and Rwanda. We haven't found any more yet, but we're still on the hunt for it. So this paper recently got accepted, thankfully, into Nature Communications. We have a triple first author to show how everyone's contributing equally. It's a great example of what collaboration can get you. We have a triple final author, myself, Sebastian, and Philip. And it's on preprint on bioRxiv if you want to go and look at it. And that's everything. I'm happy to take questions.
That was excellent. I'm sure there's a thunderous applause from everyone at home. Don't switch your mics on to clap though, it's fine. And we have a lot of questions that came up during the session. What time is it? We got 5:30. Okay, and we'll go through a couple of them and then we'll have to move on. So
Yeah.
one of the first ones is well, I won't ask my own question, I'll ask other people's questions. So when you look for monophyletic groups in taxonomy, the choice of your gene, SNPs, MLST has a big influence. How do you decide what characteristic you to use to build a taxonomy of Mycobacterium tuberculosis?
So these days, especially for TB, we will always use the genomes because the three different types that are often used for this is the Spalago type, which I showed has a lot of convergence inside of it. So these patterns can converge across the tree and are not very good for building this. Naki Komos published on this a few years ago. We have VNTR, it's called Miri VNTR, also has some convergence, can give you these larger groups, but overall we're using genomes for everything these days. And I think for a lot of bacteria, that's the way to go now. I'm very much a fan of extra information means extra resolution.
Well, I mean, a follow-up from that I had was that you do mention that you had to exclude lepre from your analysis on the basis that you wouldn't get enough whole genome. I mean, isn't that not a little problematic?
So I had originally included it with lepre, and it just gave me such a small core alignment that I could, let's say, put that into the paper and said, look, we've shown that this root is still true, but based on a couple of hundred genes. Whereas if I took out lepre, then the core genome went up to about 1,500 genes. So it just gave me more resolution that I was able to really make sure that this rooting was correct. When we did the entire phylogeny of the entire genus, we had everything, including lepre, ulcerans, all these reduced genomes in there. So it depends on the question you're trying to do. I just don't put the reduced genomes in when I'm trying to outgroup it because it gives me less resolution.
Okay, I'll ask one quick other one. Given that you use packed biosequencing, is there any chance that you or your colleagues have looked at the DNA methylation profile and compared it to the strains from other lineages?
No, I know Jody Phelan published on the methylation a few years ago, and I haven't done that myself, but we have all the raw data, so I'm sure we could go back and do that, and that might be something that we will do. We're hoping to follow this up with some mouse studies to look at the virulence, and I think that maybe can go in with all that, which is a great suggestion. Thank you to whoever said that.
Excellent. So whoever asked that can get in touch with Conor and see if you want to help out.
Yeah, for sure, because it's something new to me, so I'm happy to have other people drop in on it.
All right, that's excellent. All right, that was really good. There's a lot of questions going on in conference room 1 on the Discord, Conor. So
great. I
will redirect you to there, and you can have a look at those. But we've sort of run out of time, so we'll have to move on
no problem. Thanks.